The structure of the DNA molecule starts with the building blocks of a nucleotide. So it's a phosphate, a sugar, and a nitrogen base. And then this is the other side of the molecule. It's the same thing, a nitrogen base, a sugar, and a phosphate. So we'll look at this model here. I have a red bead that represents the phosphate group. The white bead represents the sugar. And for DNA, the sugar's called deoxyribose. And for RNA, the sugar is called ribose. And then there are 4 different nitrogen bases. A is for adenine, it's yellow. G is for guanine, and it's green. T is for thymine, and it's orange. And C is for cytosine, and it's blue. The 2 A and G's represent the purines, and the 2 T and C's, they're only single ringed structures, carbon rings, and they are pyrimidines. And so they're the smaller one. Okay, and then the 2 structures are held together by a hydrogen bond. It looks like a little miniature dumbbell, if you can tell from my sticky stuff behind it. So the building blocks of DNA and RNA include one of these 4 nitrogen bases and then all these other parts. So we have a phosphate bonded to a sugar in this way, and then the nitrogen base is A bonded off the sugar this way. So the sugar is kind of the pivotal molecule. So that's what a nucleotide is made of. So it's got a phosphate group. It has a sugar. And if we're talking about DNA, deoxyribose, RNA is ribose. And then this is the nitrogen base, which could be one of 4. And if we're talking about DNA, it's one of these 4. If we are describing an RNA nucleotide, it has these same building blocks, phosphate, sugar, and a nitrogen base, but it has, instead of a T, it has a U for uracil. So if you could look up here at my screen, I have the 4 nucleotides here. This is DR for deoxyribose, for DNA, and the phosphate group. And then this is the carbon chain. This is the C, the carbon chain for cytosine. And this also is a deoxyribose with a guanine. There's the 2-ring structure of a purine and the phosphate group over here. So that's these are DNA nucleotides. These are also DNA nucleotides. And for a RNA molecule, this is a phosphate group bonded to a ribose instead of a deoxyribose, and there is a uracil. So if you look at the ribose sugar, there's an OH group on the 3rd and the 2nd carbon. In the deoxyribose, there's only an OH group on the 3rd carbon and not the 2nd carbon, so it's deoxidized. And that's what makes it deoxyribose. Then over here it shows too the uracil, the U, doesn't have a carbon bonded off of the side of it, but the T does. The T goes with DNA. So there's the missing oxygen on DNA, and there's the oxygen on the RNA molecule. So those are the differences between the 2 nucleotides. The other difference, one more difference there, shows the 4 bases that are in DNA and how they are double-stranded. Where the RNA has its 4 bases, it has uracil instead of thymine, and it's just a single strand. RNA is typically smaller, only 1,000 or so bases, whereas DNA is billions. So if we were to build a DNA molecule, we would take a nucleotide like this. There's the sugar, phosphate, and nitrogen base. And then the other nucleotide that binds to the other side of it, also phosphate, sugar, nitrogen base. and they're held together by a hydrogen bond. So this represents a base pair. And the base pairs are held together by the hydrogen bond. RNA doesn't bond double-stranded, only temporarily. And then if we just continue to build the chain here, so if I added another base pair to this model, you can start to see a ladder forming. So what we get then is this structure. This is the straightened out ladder of DNA molecules with what Rosalind Franklin described as the sugar phosphate backbone. Phosphate, sugar, phosphate, sugar, phosphate, sugar, and it alternates as the same over here. And then all of the bases, the G bond with C, C bond with G, A with T, A with T, C with G, and so on. It's always the same. So that's called the base pair ruling that the DNA molecule uses. A always binds with T, C always binds with G. So then when we throw the RNA structure in there, RNA temporarily binds to DNA. So if A is on DNA, let's write a DNA strand here, A, T, C, and G. So if RNA comes in here to temporarily bind to it, the A binds with the U instead. And if there's a T on DNA, RNA has an A, C binds with G, and G binds with C. So the U is what's different in the sequence. And then what's not in the drawing here is the sugar's different too, without that oxygen there. So those are the comparisons of the structure of DNA. Now put that all together and twist it. The twist is the helix part. The double is the two strands, so there's the double helix of the DNA molecule. Okay, the last part is the function of DNA and RNA. So DNA is the storage molecule, so it's huge, over 6 billion nucleotides, and it also is transferring genetic information, so it could be copied and shared in the new cells as mitosis and meiosis occur. The RNA molecule is smaller, only thousands of base pairs in size, or bases, and it codes for these amino acids or these proteins by acting as a messenger between DNA and the ribosome to make proteins. So it's the runner. It carries the information from one place to the other, translating it into the protein. So those are the functions of DNA and RNA. And then an important note to make is we're making proteins. DNA codes for proteins. All enzymes are proteins, and enzymes control cellular metabolism. So everything that a cell does, chemically speaking, at the chemical reaction level, requires an enzyme to run it. So whatever enzymes you have determines what kind of chemical reactions you can undergo. So that's what makes the DNA molecule such an important structure in our cells.